Hello everyone. So today let's start a new chapter, that is molecular basis of inheritance. Okay, here when we are studying about the molecular basis, that means here we will study about the nucleic acids, DNA, RNA, the genetic materials, how they are transmitted, what are the structures. Okay, all these things we will study in details. How the DNA replication takes place, RNA transcription, protein synthesis, all these we will study here step by step. So now first of all, let us study what is actually nucleic acids. Now nucleic acids, these are the main substances or the main hereditary substances which are present in a cell. It is of two types, the DNA and the RNA, isn't it? Now in our earlier part, that is in maybe in your school levels, you have studied about what is DNA and what is RNA. So DNA, that means it is deoxyribonucleic acid. It is the genetic material of the cell, and this was first of all identified by or discovered by Frederick Miescher. Okay, now the DNA, it occurs in almost all cells, isn't it? Now as it is the genetic material, so it has to be present in all cells. In the prokaryotic organisms, the DNA is a naked DNA. It is circular DNA, which we call it as plasmid also. Okay, it is embedded in the cytoplasm. Often it is known as the nucleoid also. So that is the DNA present in prokaryotes. Now in eukaryotes, the DNA is linear. Okay, it is confined to the nucleus as a component of chromosome, and it is known as the nuclear DNA. That means here the DNA is circular, here the DNA is linear. Okay, next, the DNA which is present, the quantity, it depends upon the cells of the species. So upon the species, the quantity of the DNA, it may vary. Now the gametes contains half the amount of DNA, but the somatic cells, it contains twice the number of DNA content of the gametes. Okay, now the length of the DNA, it is actually the long polymer of deoxyribonucleotides, and the length is determined by the number of nucleotides that are present, or a pair of nucleotide referred to as the base pair which is present. Okay, so the length may vary from species to species. In bacteriophage, it has about 5386 nucleotides. Again, in haploid content of human DNA, that means in gametes, it is 3.3 into 10 to the power 9 base pairs. Okay, in E. coli, it has 4.6 into 10 to the power 6 base pairs. So accordingly, it will vary. Now, we are telling that the length of the DNA is determined by polynucleotide chain. So what is this polynucleotide? The polynucleotide, poly, we know poly means many. Nucleotide consists of three parts: nitrogenous base, pentose sugar, and the phosphate group. Now the nitrogenous base, it is again divided into two parts, a purine and a pyrimidine. Now purine is a double ring structure. Okay, and here the nitrogen is at the position 1, 3, 7 or 9, and it is again subdivided into adenine and guanine. The pyrimidine, which is a single ring structure, here nitrogen is at position 1 and 3 only, and it is again divided into cytosine and thymine. But what happens in case of RNA, the thymine is replaced by uracil. Okay, so as we are studying about here polynucleotides, so we will get all these three. So pyrimidine, either cytosine, thymine or uracil. But always remember, in DNA thymine is present and in RNA uracil is present. Then pentose sugar, that means it will be a 5 carbon sugar. Okay. Then it will, in DNA it is deoxyribose, and in case of RNA it is only the ribose sugar. And phosphate group, it is present. Now if the nucleotide, we are telling it consists of three types: nitrogenous base, three components, pentose sugar, phosphate. This together constitute the nucleotide. But if nitrogenous base and pentose sugar is present, then this particular structure is known as nucleoside. That means SIDE. This nucleoside word is used for these two structures. Okay. But if phosphate is present, then we will use the term nucleotide. Now, the polynucleotide chain, it consists of different types of bonds. Now, when we are studying about a polynucleotide chain, it consists of, first of all, the glycosidic bond. That means the bond between the nitrogenous base and pentose sugar. Now, we are telling that these are interlinked, isn't it? These three components are linked together to form the nucleotide. So, the main bond between the nitrogenous base, that is adenine, guanine, cytosine or thymine, with the pentose sugar, that particular bond is known as the glycosidic bond. Again, the sugar is attached to phosphate, or else we can say that the phosphate is attached to two sugars on either sides. So that particular bond is known as the phosphodiester bond, which is the main bond or the backbone formed in case of the DNA structure. So sugar, phosphate, sugar, this particular bond is the phosphodiester bond. Now again, when we will study the structure of DNA, then we will get the hydrogen bond also. That means the two polynucleotide chain will be interlinked with the help of hydrogen bonds. But basically, for a single polynucleotide, we will get only these two types of bonds, the glycosidic bond and the phosphodiester bond. Now, next we come to the term polarity. Polarity of polynucleotide chain. That means while we are studying the structure of DNA nucleotide, here what happened in the chain, one end it consists of a free phosphate moiety at the 5' end of the sugar, which is referred to as the 5' end of polynucleotide chain, and simultaneously on the other hand the polymer, the sugar has 3' hydroxyl group, and it is referred to as the 3' polynucleotide chain. That means if we are drawing a polynucleotide, if one end is 3', definitely the other end is 5'. So that is known as the polarity of the nucleotide chain. Okay. So these are actually the basic concept, basic introduction regarding the DNA structure. So next we will study about the DNA structure in our next class. But before that, we have studied what is it actually. It is the genetic material, nucleic acid, which was first discovered by Friedrich Miescher. It occurs in all prokaryotic and eukaryotic cells. The basic component, it consists of polynucleotide chain. So how the nucleotide is constructed with the help of nitrogenous base, pentose sugar and phosphate. How they are divided, each and everything is important. So go through this. This chapter is very important as the Mendelian principles. So go through them, go through the introductory parts very well. Then only you will be able to understand the further parts. So if there is any problem, let me know and go through the videos and the books properly.